Namaste to all students. Kusha Vidya, myself, JV and Dr. Kushbu Verma from Faculty of Agriculture and Veterinary Science. Today we are going to discuss about secondary metabolites. So first of all, you have to know about what is secondary metabolites. So secondary metabolites are generally defined as small organic molecules produced by an organism that is not essential for their growth, development, and reproduction. They may include pharmaceuticals, flavors, fragrance, food additives, and feed stocks, etc. Why plant produces secondary metabolites? This is a major question. So plant hormones, which are secondary metabolites, are often used to regulate the metabolic activity within the cell and oversee the overall development of the plant. So it protects plant against herbivores and microbial pathogens, means it is involved in the plant defense mechanism, and it serves as attractant for the pollination and seed dispersing animals. There are various types of the secondary metabolites like alkaloids can be present, cyanogenic glycosides, flavonoids, phytic acid, gossipyl, and phytoestrogens and carotenoids. So if we are going for the type of alkaloids, so there is example like caffeine, codeine, and quinine. So they are used for as a stimulants, analgesic, and anti-malarial. And second type is cyanogenic glycosides. So example is diogenin, which is also used for the progesterone. And if we are talking about the flavonoids, so examples are quercetin and procyanidins, and they are used as an antibacterial, antioxidant, and anti-inflammatory. And phytic acids are used as antioxidants. And gossipol is basically hypokalemic paralysis attack, and phytoestrogens are basically used for reduction of risk of cardiovascular disease, and examples are resveratrol. And next type is carotenoids. So examples are alpha carotene, beta carotene, and lycopene. So they are basically used to contribute to photosynthesis process. So there are major types of secondary metabolites. If we are talking about the hormones, so abscisic acid, auxin, cytokinin, ethylene, and gibberellins are also secondary metabolites. So abscisic is basically inhibit growth in response to change in temperature and light. It control closing of stomata in dry condition. ABA is applied on plant before shipping. And second hormone is auxin. So it stimulates the cell elongation, differentiation of xylem and phloem, root initiation, suppresses growth of lateral buds, delay leaf census. And next hormone is cytokinin. So it promotes cell division, lateral bud development, and delay of census. And next hormone is ethylene, which is used for the fruit ripening and inhibition of stem elongation. And next hormone is gibberellin, which is used for the synthesis in apical portion of stems and roots. It is important. It is having important effects on stem elongation. Secondary metabolites, why in vivo production? So according to the WHO survey, approximately 70 to 80% of world's total population depends on herbal drugs. Some compounds are difficult to synthesize chemically due to its structural complexity. So some novel compounds produced in cell culture are not produced in intact plant. At least 85 novel compounds, including 23 alkaloids, 19 terpenoids, 30 quinones, and 11 phenyl compounds have been isolated from some 30 different plant culture systems. So there are two methods of in vitro culturing, callus culture and suspension culture. In process of suspension culture, you have to create callus culture. So first step to establish cell suspension culture is to raise callus from any explant of the plant, and to maximize the number of or production of a particular compound, it is desirable to initiate the callus from the plant part that is known to be higher producer. And calli are generally grown on the medium solidified with gelling agent. In morphological terms, it can vary extensively, ranging from being very hard, compact, or green or light green in color, where the cells are extensive and strong cells to contact, cell to cell contact, and friable when the callus consists of a small disintegrating aggregates of poorly dissociated cells and has brownish or creamy appearance. There are various lists of products prepared by different cultures methods. So like alkaloids you can generate by the suspension method, allicin from callus culture, and caffeine from the callus culture, and triterpenes from callus culture you can generate. Suspension culture could be run as batch culture or continuous culture, and it is basically predominantly batch culture is used because of many secondary products are not growth associated, and genetic instability of the cultured cells, and operability and relatability and economic consideration also suggest for the use of batch culture. So the growth of cell suspension culture with respect to the time is best described by the sigmoid curve. The growth rate is measured by the steepness of the curves, and it is the steepest when the population density reaches one half of the carrying capacity. As we know, there are two types of suspension culture, batch culture and continuous culture. So batch culture's sub-examples are slowly rotating culture, shake culture, spinning culture, stirred culture. On the other hand, in the continuous culture, there are two types of a culture like chemo states and turbido states. In slow motion rotating flask, in this culture, single cells and cell aggregates are grown in a specially designed flask, the nipple flask. Each nipple flask possesses 8 nipples like projections having a capacity of 250 ml. They are loaded in a circular manner on the large flat disc of vertical shaker. When the flat disc rotates at a speed of 1 to 2 rpm, the cells within each nipple of the flask are alternatively bathed in the culture medium, and that's how this rotating culture can be maintained. In second type, which is shaker culture, so it is very efficient system. In this method, single cells and cell aggregates in fixed volume of liquid medium are placed in conical flasks. These flasks are then mounted with the help of clips on a horizontal large square plate of a orbital platform shaker. The square plate move in a circular speed of 60 to 180 rpm. Next type is spinning culture. So in this culture system, large bottles are used, usually with a capacity of 10 liter. Large volumes of cell suspension is cultured in 10-liter bottles, with the bottle spinning in a spinner at 120 rpm at an angle of 45 degrees. In exterior culture, this system is used for large-scale batch culture. In this method, the large culture vessel is not rotated, but the cell suspension inside the vessel is kept dispersed continuously by the bubbling sterile air through the culture medium. Internal magnetic stirrer is used to agitate the culture medium safely. The magnetic stirrer revolves at 200 to 600 rpm. Next type, which is continuous culture. So in continuous culture system, the old liquid medium is replaced continuously by the fresh liquid medium to stabilize the physiological state of the growing cell. In this system, nutrient depletion does not occur due to the continuous flow of nutrients, and the cell always remain in the steady growth phase. Continuous culture is further divided into two types. First one, enclosed type, The used medium is replaced by the fresh medium. Hence, the cells from used medium are mechanically retrieved and added back to the culture, and thus the cell biomass keeps increasing. In open type, both the cells and used medium are replaced with the fresh medium, thus maintaining culture of at constant and submaximal growth. And open continuous cell suspension culture is of two types. Chemostat. In this system, culture vessels are usually cylindrical or circular in the shape and possess inlet and outlet pore for the aeration and introduction and removal of the cell and medium. Such a system is maintained in a steady state, and second type is turbido state. So a turbido state is a continuous culturing method where the turbidity of the culture is held constant by manipulating the rate at which medium is fed. In this system, the cells are allowed to grow up to a certain turbidity when the predetermined volume of culture is replaced by the fresh culture. The turbidity is measured by the changes in the optical density of the medium. So that's how you can create your cell suspension culture. So this is the sum up of this session, and that's how you can create the secondary metabolite by cell suspension culture. And this session is powered by digital version 2.0 Jyoti Vidyapeeth Women's University Jaipur. If you have any query, then mention in the comment box. I will resolve it. Thank you.